Thanks very much, Dave. Okay, so hi everyone. As Dave said, I'm going to be talking about some of the work that we're doing in Birmingham around sperm, and particularly the way in which sperm generate a beat that will hopefully progress them from their really complicated internal apparatus. So as Dave said, this is kind of collaborative work between me and him, and also Jackson Kirkman Brown at the Women's Hospital as well. So the data that we've got comes from donors over at the Women's Hospital. So that's great. So the motivation for this work is really the problem of infertility, in particular the problem of male infertility. So infertility is a widespread problem worldwide. So 1 in 6 couples fail to conceive after a year of trying for a baby. Historically, this has often been thought as a female problem, treatments very much focused on the woman. But we now know that half of fertility issues come from some male problems, some male factor. And obviously the big component of male fertility is the sperm and the ability of the sperm to migrate through several different types of fluid environment, propelling itself with a single tail that beats side to side, as you can kind of see in this little plot in the top right. And we've got to be able to progress through the female to the egg, penetrate the egg, and hopefully fertilize. Treatments for infertility pretty much boil down to taking some form of ART, some form of assisted reproductive technology, such as IVF. These techniques tend to be very expensive. They tend to be very invasive, particularly for the female partner, and they carry a lot of risk. In terms of treating problems to do with male infertility, you're really looking at just a treatment called ICSI, which is very similar to IVF, where eggs are removed from the female partner, but a single sperm is then chosen and injected straight into the egg. So you're kind of removing that motility component from the problem. However, the way that sperm swim and the ability of sperm swim is a real important kind of natural selection, where we can kind of get rid of the sperm that can't swim, that can't progress, potentially have issues with maybe their DNA packing or the way that their proteins have been created. Maybe they're not being able to generate the waveform that we expect. So actually understanding the kind of propulsive aspect and the physical aspect of this is a really key important part of what's going on. So I've already mentioned that the internal structure of flagellum is really complex. If you've ever done any work on sperm or any other flagellates or cilia, you'll know that there's this really interesting mechanical component to the internal structure. And that's kind of coupled with the biochemistry and everything else that's going on to really form this beat. I'll talk more about exactly what's going on, or exactly what the components are in a bit, but basically what we're trying to do is abstract all this detail into a nice simple model that can help us understand where the beat comes from, how the beat is generated, and therefore understand maybe problems with the beat, or maybe just be able to create a model that can fit an experimental beat and therefore allow us to probe more deeply what's going on. And as you can see in this little schematic at the top, sperm can do lots of different things. They have a really wide variety of behavior. So really we want to know, can we generate a model that is still simple, but actually explains and characterizes all these different behaviors? And can we understand how one set of structures really give rise to all these different types of waveforms? So this variety can be seen in a lot of ways. I'll give you a few examples now. So there's many things that can change and affect what's going on in the sperm. Particularly the most obvious thing might be, particularly for this seminar, the fluid environment. So in the biologically active fluids, SIG seminar, fluid environments, obviously something a lot of us are interested in. Here we've got 2 examples of some experimental cells. One on the left is in a low viscosity fluid environment, one is on the right. Or say low viscosity, we are still in a very low Reynolds number, so a Stokes flow regime, because of the very small length scales. Sperm length is in humans about 50 to 60 micrometers, but we can still have a low viscosity fluid and be within that regime. So you can see this low viscosity cell has this big side to side motion, large amplitude of beating that's kind of spread out along the tail, nice and smooth. The path tends to be quite side to side. You get a kind of yawing behavior. But then sperm tend to swim progressively in a low viscosity environment. In the high viscosity environment, you can see a real change to the waveform. So close to the head, you've got much less curvature, much more tightly packed waveform with increased curvature right at the end. And sperm that are swimming through with this sort of waveform tend to swim much more progressively, much less side-to-side movement, almost as if they're drilling through a highly viscous fluid environment. Species has a real impact in what's going on. In this talk, we're only going to talk about human because we're really interested in human fertility. But just as an aside, here's some mouse sperm, some tanquid goat sperm that have been imaged by one of our collaborators in South Africa, Professor Gerhard van der Horst. So this is a mouse sperm on the left. So mouse sperm, you can't quite see in this picture, but have this really asymmetric head with this big hook-like structure on the end. So their flagellar beat is affected by this, and they have a very asymmetric flagellar beat. They also have a very long flagellum, kind of 100, 120 microns long. Tanquid goat, on the other hand, you can see is much more symmetric side-to-side behavior. It's much shorter flagellum compared to the mouse, shorter even than humans. So this species can have a real impact in what's going on. And the other thing is the biochemistry. So an important part of sperm motility is the ability to hyperactivate. So this is a change in motility that occurs in order to penetrate the egg. And this is characterized by a real increase in bending, an increase in curvature along the flagellum, and often a loss of progressive motility. So like these ram on the right-hand side, where you have what is called this kind of star spin pattern. The cell isn't progressively swimming, but it is moving around on the spot. You can often get patterns like this, or kind of figure-of-8 patterns as sperm swim with hyperactive motility. So really, these are the three types of motility we're going to think about, and I'm going to refer to them as kind of low viscosity, high viscosity, and hyperactivated waveforms, essentially. But these are the type that I'm referring to. So kind of big picture, what are we hoping to achieve, not just with this work, but more generally at Birmingham? Well, we're really coming at motility from a kind of mechanistic point of view, a physical point of view. And with this work, we're trying to provide a mechanistic explanation of how this variety is emerging, and really improving our understanding of how sperm swim, but also what can go wrong with sperm. So obviously, if we're looking at patient samples in the clinic, we want to be able to understand them in the context of what can go wrong, and particularly how could we then think about treatment. And ultimately, with all this modeling and simulation, we're going to also be comparing with experimental data. And this FAST logo on the right-hand side is the image analysis software that we've released over the last couple of years that's free to access, that will do live tracking of sperm cells from videos of negative phase microscopy. Okay, so let's move on to thinking about the modeling and the maths. So this is the sperm. This is what we're talking about. So we've got a ellipsoidal head. We're going to discretize the surface eventually. So we're going to call that surface DH. We're going to say our sperm is at an angle phi to the horizontal. And then sperm have a single flagellum attached to a point x0 to the head. And we're going to really be thinking about a 3D head, but a planar flagellum. So our flagellum is going to be located entirely within the plane. So we're going to think about it as a 2D structure. So we can describe it in terms of an arc length S, which is going to start at 0 at the neck and go to L at the length of the tail. And then the tangent angle theta as we move along this arc length. So we've hinted a bit at the complicated internal structure, and I think many of the people in this call will be aware of this, but just to talk it through. So sperm and all flagellates and ciliates have this axonemal structure where we have an array of doublet microtubules, and in sperm we have a central pair, so this is called the 9+2 structure. We have 9 pairs of doublet microtubules, and then the central pair, the +2. But the sperm structure isn't constant all the way along its length. So we start close to the head with some accessory structures that really reinforce the cell and kind of really try and limit movement to within this bending plane. We have a fibrous sheath that's surrounding the axoneme, some outer dense fibers that also provide some reinforcement. As we move along to kind of the middle of the principal piece, we lose some of these outer dense fibers, so the stiffness is actually changing along the length of the tail. As we move further down towards the end of the flagellum, we lose these accessory structures entirely, and we end up with just plasma enclosed membrane surrounding the 9+2 axoneme. This end piece, this dotted end piece that I've got indicated at the end, is a really interesting component of the flagellum. So here we actually lose that structure of the axoneme, and we lose that kind of doublet arrangement, and we end up with just these singlet microtubules. Essentially, the end piece is a space where the microtubules can kind of slide past each other with no active component to bending, which allows the bending to happen in the rest of the flagellum, essentially. It turns out that this end piece is actually really useful hydrodynamically. We've got a paper by my PhD student with Dave, Cara Neal, released last year in Physical Review Fluids, where she showed that actually this end piece confers a significant advantage to the cell, allows it to swim faster and allows it to swim more efficiently than if the whole of the tail of the same length was actively bending. So that's really cool. So this is a really complex amount of structure. It's there's a whole lot going on between these doublet microtubules. We have these little dining arms are walking between neighboring pairs and pulling on each other, which is what induces bending. There's a whole load of biochemistry going on and signaling that's going on that's controlling everything that's happening within the cell. So really, without wanting to model all the little details going up, we want a way to kind of abstract all this away into a simple control mechanism that we hope is going to give rise to some sort of beating. And this idea isn't new. People have talked about it previously, particularly going back to kind of the 1980s, maybe the late 70s with Brokaw's work, where he proposed a model of curvature control, which basically says that the flagellum wants to bend, all the internal apparatus wants to bend the flagellum up until it's bent a certain amount. Once it's bent too far, can no longer sustain bending, and it's going to start bending in the opposite direction. Brokaw did a great bunch of work on this, a whole series of papers that were really interesting, but eventually concluded that maybe there were some problems with the curvature control model and that it was incompletely specified to be able to show all the beating styles that we see in nature. So this led to Lindemann's hypothesis of geometric clutch, which is basically the idea that bending is introducing a tension. And if we bend too much, the forces in the bending region are essentially going to cause the dyneins to disconnect, which is the clutch mechanism, which is going to cause bending in the opposite direction. And then Ulica and colleagues came up with a model of sliding control, whereby they coupled the motor activity to the sliding of two passive filaments next to each other, which would then induce bending. So these models have all had some success in creating different aspects of flagellar beating, but particularly we're really interested in working out something nice and simple, really taking a step back and seeing, can we get something that we can fit to our experimental data and that gives us all this insight. So we're going to take this idea of curvature control, but come at it from a more mathematical approach, but also with all this structural information that we've developed, such as the change in elastic stiffness along the flagellum, the inclusion of the end piece of 3D non-local hydrodynamics for the cell, and see if that's going to help us show what's going on. So I've said we're taking this curvature control approach, this 3D non-local fluid mechanics, and a realistic but planar geometry. I can talk more in the questions if you want about the kind of planar assumption, but for now, as a first model, this is pretty reasonable. So this is our simple active curvature model. Then what are we looking for from this model? Well, essentially, we're having on any section of the flagellum some internal moment per unit length that's going to be the culmination of all this internal mechanisms and internal structures acting to bend the tail. And it's going to want to bend the tail with some preferred moment density, we'll call it MC. So this is going to induce bending. If everything's working together, it's going to be working together with this moment density MC. And then at some point, it's going to reach some critical curvature threshold, which is a parameter in our model, and it's going to want to disconnect and bend back in the other way. So just schematically then, we can imagine that we're bending with this preferred density MC. That's increasing curvature. We reach a threshold. That induces bending in the opposite direction, and then we can create some sort of limit cycle. And we can imagine that anywhere off this limit cycle, we're going to want to kind of pull ourselves into this limit cycle. We're not going to have instantaneous switching between one direction and another, because there's a lot of stuff going on internally. One pair of dyneins may disconnect, but this neighbor pair may still be connected. It's not an instant switch, but this does kind of capture the ideas that we're trying to convey. And really, we're going to wrap this all up in this nice little ODE that I've written on the left. So we have some switching time toward D, multiplying the rate of the time derivative of our moment density m. And essentially all that this is saying is the image on the right-hand side. So we have a Heaviside function that dictates which side we're going to switch. And we're trying to be attracted to either MC or minus MC, depending on which side we're on. And the idea is that we couple this together with fluid dynamics, some elastohydrodynamics that explain the tail. And this should all come together to fit into our model and hopefully develop a simulated cell. So this is how we're going to model the cell. So we're going to use a method that we've been developing over the last couple of years, which is an integral operator formulation of the elastohydrodynamic equations. So this comes from work by Moreau et al. In 2018. And then another one of my PhD students, Asketul McNair, published on this, where he developed the non-local extension of this. And we're going to couple this elastohydrodynamics of a flexible filament, essentially a 1D filament, which is our tail, to the head using the regularized Stokes-Slit method, and particularly the nearest neighbor version of the regularized Stokes-Slit method that we've been developing in Birmingham over the last couple of years. So we discretize our tail into some straight line segments, and these are the blue lines you see in the picture. Each segment has a tangent angle theta, and then that goes across the tail from s equals 0 all the way to the end. We have some force densities. So the force densities along the surface of the head, we're going to denote by phi. So those are forces per unit area. And then F, the forces per unit length, which are going to be constant along each segment of the tail. And then the curvature we can calculate just by taking finite differences of these tangent angles theta, and then across the head with the head angle phi in the first theta on the tail. We said that the elastic stiffness is going to vary along the flagellum, and as we saw at the start, essentially we have these thinning accessory structures over the first section of the tail, and then when these accessory structures go, we've just got a constant 9+2 axoneme, so we kind of go to a constant stiffness after some distance d. So this parameter d is essentially the length over which the elastic stiffness varies. We have ED, which is the distal stiffness of the flagellum, and rho, which is the ratio between the proximal stiffness and the distal stiffness. So we have quite a good experimental approximations for ED, coming from measurements of things like the bear axoneme and sea urchin flagellum that don't have these accessory structures. But really, we're going to keep that ratio rho in the model as a parameter that we can kind of play about with, because we don't have a lot of good experimental measurements for exactly how stiff the tail is at the neck. So Deb has just asked a question that I think I will answer in a second as I talk through. So if you bear with me, we can answer that as we go, hopefully. And then, as I've said, we're using the regularized Stokes-Slit method. So we have our regularized Stokes-Slit, which is the Green's function for the Stokes equations that are forced by a point force that's smoothed out. So our system of equations, so we're non-dimensionalizing with respect to some scales. So L, the length of the sperm, MC is our preferred moment density. We've got fluid viscosity mu, our distal stiffness, flagellar stiffness ED, and then that time scale tau D. And then this gives us our non-dimensional system of equations. So we have force-free and moment-free swimmers, because we're thinking about a sperm that's swimming in an infinite expanse of fluid. We're not going to consider the effect of any boundaries. Again, I'll talk about that a little bit towards the end, but for now, we're not thinking about boundaries. We've got our integral equation for the elastics, which is essentially balancing the curvature of the cell with the internal active moment m. This Heaviside function here is a switch to essentially turn off that end piece, so we don't get any active contributions from the end piece. and then the moment induced by the hydrodynamics of the surrounding fluid. And then we have our equations for the hydrodynamics of the cell with the head and tail taken into account. And then obviously this ODE for our internal active moment. So we have three dimensionless groups here, M, S, and kappa C, and then we have our dimensionless parameter rho, which is the ratio of elastic stiffnesses. So these are our four parameter groups. So M is the ratio of essentially the active forces to the elastic forces. S is kind of like a sperm number. It's the relationship of the viscous to elastic forces. And then kappa C is obviously the dimensionless critical curvature, and rho, again, the ratio of elastic stiffnesses. So we can write this system as a big monolithic matrix system, AV equals B, for some unknowns. So the unknowns being the position of the cell, the head angle, the tangent angle of each section of the flagellum, the force per unit length over each section of flagellum, and the force per unit area discretized over the surface of the cell head. And we can write this as a reduced ODE for just the rate of change of position, direction, and tangent angle, and then solve this using one of MATLAB's built-in ODE solvers, which is a really nice function of the method that we've set up. And there's one more kind of technicality of implementation, is that we've also imposed some sort of near interaction force, some sort of repulsion force. This is just to kind of ensure that the flagellum doesn't kind of tie itself up or cross itself as it bends, as the beat is evolving. This only comes into play when the cells or two segments are very, very close to each other. And we choose this nice form by Ishikawa and Pedley that we only implement when two segments are very, very close to each other. And it turns out once cells are swimming, this actually doesn't come into play. It's only when the beat's kind of slowly developing that this is actually useful. Okay, so that's kind of a whistle-stop tour of the setup. So let's talk through what we see when we simulate this problem. So for each simulation, we select our parameter sets. We initialize one cell on its own, no boundaries, just infinite expansive fluid with a constant active moment across the tail and some shape that's some sort of low amplitude parabola. And we run the simulation until a periodic beat is achieved. And then we can compare and see what's going on. So obviously, the first question is, well, can this model, this nice simple model, can it drive a progressively swimming beat? Can we induce swimming and induce something that looks kind of like we expect a sperm to see? So here's an example. The answer, luckily, is yes, we can drive a progressively swimming beat. We can do something that kind of looks like a sperm. It's got a traveling wave progressing down the flagellum, as you see on the left. It's tracing out a path that, if you squint, looks kind of like a sperm beat. We can believe that this is the simulation of a sperm. I've color-coded the segments of the tail here, and on the right-hand side, the colors match the segments of the tail, and we're showing the active moment against the curvature of the flagellum at that point. So here you really see the nice limit cycle behavior that's kind of occurring. The dotted line here are the preferred moment and preferred curvature. And we see that the exact dynamics of the limit cycle really depend on where on the flagellum we are, what the interaction is between the changing elastic structure and the hydrodynamics and that internal moment ODE that we specified. So that's good. This would be a pretty short talk if we couldn't drive a progressively swimming beat. So let's have a think about what's the variety then. This is the big key question. Can we replicate what's actually going on in the world of sperm? So here we've fixed two parameters. We've fixed S and we've fixed rho. And we're varying M, which is the amount of active moment essentially that the sperm's inducing on itself, and kappa C, which is the critical threshold. And gaps in this plot are basically where we don't get a nice stable beat that's periodic or stationary. So we can see quite a number of different behaviors here. If we start at the bottom, we have something that to eyeball norm, looks very much like what we might expect a high viscosity sperm beat to look like. We move up, we get some sort of rapid change to something that looks maybe like a low viscosity beat, so wider, increasing curvature, side-to-side movement, greater amplitude of beating. And as we move up to the kind of high KCs, high kappa Cs, we get something with greatly increased curvature, something that might look more like the kind of hyperactivated cell that we saw near the start. So this is encouraging. We're really showing that there is a variety of behavior coming out of this very simple model. But there's also some interesting things going on. So a big question about what's happening in this kind of step change here. Can we make a bit more sense of that? And then this kind of slowly varying step between these kind of middle beats and these hyperactivated, quote unquote, hyperactivated beats at the top. And that's something that we can probe a bit more. And also let's have a think about what do the tracks look like? Are these cells actually progressive, or are they just making something that might look sperm-like but actually isn't? So this is the same data, but this time we've plotted the tracks in red and then the cell head at the final point. So you can see, as I described at the start, the kind of hyperactive, high viscosity beats have much decreased amplitude in their path, and much straighter paths, much more progressive. These kind of low viscosity beats, much more side-to-side behavior. And then we're actually losing the progressive nature of the beat as we move up here, get into these kind of figure-of-8 motions that we talked about right at the start. So that's interesting. That's certainly describing the sort of things that we'd see in cells, even down to the beats that aren't quite evolving on the left-hand side. I'm not seeing anything here that we're kind of looking at and saying definitely isn't a cell. So we're not getting a whole load of parameters that we're saying aren't reasonable. So that's encouraging as well. So let's think a bit more about what's going on in this sharp change between what I think of as a high viscosity beat and what I think of as a low viscosity beat. Let's really think about this switch in behavior. So we can think about looking at this just qualitatively at how the waveforms look, or we can think about something slightly more quantitative, and we can think about the average swimming velocity for these cells. And if we look at the average swimming velocity, so this is the same plot, but color here is swimming velocity. There really is two smoothly varying regions with a real step change between them. And this is much clearer in the 3D plot of the same data, where we see this real cliff edge between the faster swimming, higher amplitude beat and the slower swimming, but more high viscosity looking beats. It's probably worth pointing out here, and this goes some way to answering Deb's question as well, the time scale for this active moment density is really encompassed in our parameter S. So it may not be that one of these types of motility is slower swimming or faster swimming, but actually it's the balance between this time scale and the viscosity and elasticity as well that's really setting what's going on. So we're not implementing any sort of natural frequency in this problem. All the timescales is kind of encapsulated within this S parameter. So this real cliff edge is kind of interesting, and I think deserves more investigation. Something that we do know is that these sperm, if we take an actual experimental sample of sperm and we image them with no stimulants, no external or weird stuff in the environment, and we put the same or sample from the same sample in each of a low viscosity and high viscosity fluids, they will do different things. So this is very much a fluid dynamic phenomenon rather than a biochemical process necessarily that's going on in the gel, or a change to mechanism. So this is quite interesting that we've got such a sharp change with little changes in parameter sets. So to investigate this further, what we're going to do is we're going to take a single parameter pair. So we're going to take m equals 900 and kc equals 4. And we're going to start just above this cliff, and we're going to move down and really try and tease out what's going on. So we'll start with this waveform, capital C equals 4, and we're going to take the shape of this waveform as our initial condition, decrease Kc, and we're going to get this waveform here. Again, we can take this shape as our initial condition, solve for Kc equals 3.4, and we can keep doing this and keep moving down until we just switch over this cliff, and you see there's a noticeable switch in behavior. So then a sensible question to ask would be, is this really dependent on what our initial condition is? So if we do the opposite, are we going to recover the same pattern, or are we going to recover something different? So let's try that. So let's start with the same waveform. And we're going to use this waveform now as our initial condition for kappa C equals 2.8. And we're going to get something that looks very, very similar. And then we can do the same, but this time we've noticed something different. So we've still got a periodic, a stable waveform that's swimming along, but actually this switch in behavior doesn't seem to occur until we get up to kappa C equals 3.7. So this very much looks like what the dynamical systems literature would call a catastrophe bifurcation, where we've got this real significant change in dynamics and having a really significant dependence on what the shape was before we go over this edge. So this is really interesting to me. This is suggesting that actually this switch between high viscosity and low viscosity waveforms doesn't require any change on behalf of the cell, but it's the interaction between the cell and its environment, which is something that we've known from experimental theoretical studies before, looking at the impact of beats with viscosity. So what about these hyperactivated waveforms at the top then? So to get to these, we kind of smoothly vary. We don't get this kind of big step change again for the kind of high levels of Kc 7, 8, 9 that we're seeing here. So how do we explain this? How does this make sense in context of the biology? Well, when cells are undergoing this hyperactivated motility, what we often see is a real increase in calcium levels within the cell. So as cells capacitate and they hyperactivate, they tend to release their calcium stores. This leads to an increase in ATP, which allows these dining arms to move faster and walk faster between the microtubules. So we suggest that this allows the cell to maybe sustain greater values of Kc, so be able to bend more before the dining arms have to release. And therefore we see an increase in Kc, and therefore we can see this hyperactivated motility. So that kind of gives us some nice insight into what could be going on within the cell, and I think a nice link between the experimental side of things and our understanding of the internal apparatus and what's going on, and what we're seeing as we look down a microscope looking at the cells. So that's okay. So sorry, just to answer Tim's question. So Tim asks, why do I say that the low Kc results are high viscosity waveforms? So I'm really just using the nomenclature high viscosity, low viscosity waveforms as reference to the typical beats that we see in high viscosity environments experimentally and low viscosity environments experimentally, just to distinguish the two shapes. I'm not actually talking about viscosity when I say that, just to try and clear that up. So it makes sense, given that we've said kind of to eyeball norm, we can see a lot of the variety that we see in human sperm in our simulations. But how does that actually stack up when it comes to comparing with experimental data? So we spent a lot of time in the last couple of years actually working out ways to track experimental cells and extract the flagellar waveform. So let's put that to use and see if we can compare with our actual experimental data. So we've got a dimensionless model. We've solved this for parameters s to the 4 in the range 12 to the 4, 18 to the 4. So 12 to the 4, 13 to the 4, 14 to the 4, etc. M between 100 and 2000 in increments of 100. Kappa C, sorry, that should be a kappa C, between 1 and 9.5 in increments of 0.5. And then 3 choices of proximal to distal stiffness ratio, just shown here. We've noticed, as I've already said, the time scale is contained within s to the 4. So that makes it slightly tricky to fit experimental data, because we really need to think about how it is we're going to account for any time variation in the problem. The other thing that we've already mentioned and already talked about is that sperm paths we know are affected by the presence of boundaries. In these simulations, we haven't included boundaries because of the sheer volume of numerical computation time that's been needed. However, we have done tests to see how the waveforms evolve in the presence of boundaries. And luckily, the waveforms themselves seem to be very similar, even with boundaries taken into account, even though the path definitely has a noticeable change. So the cells that we're imaging in 10 micron deep chambers. So it's essentially 2 parallel plates separated by 10 microns in length, with cells swimming somewhere between them. So we don't know exactly where the cell is in those images, but we do hope that these won't have a massive impact on the waveform, even if it does change things like the swimming velocity and the path. The other thing we need to think about is errors, and errors particularly in the experimental tracking of cells. So as cells are swimming, they don't tend to line up perfectly with our imaging plane. It would be nice if they did, but they don't. So we have cells coming in and out of focus. Obviously, these are very, very small, rapidly moving creatures. So we need to image that, and there's some error associated with that. But also the end piece of the sperm, and particularly the end of the tail, is very, very thin as we lose these kind of accessory structures and other things that make the tail thicker. So actually, imaging that can be quite difficult, and particularly the end piece is very, very hard to see. So we need to think about how to deal with that as well. So the fitting procedure that we're going to use, and particularly the metric that we're going to use, is the idea of mean absolute curvature along the flagellum. So we're going to start at s equals 0, go towards s equals L. For each segment of the flagellum, we're going to calculate the mean absolute curvature over a number of beats for both the experimental and simulated cells, and use that as our metric for comparison. But the thing to note is that we have a full length of flagellum for our simulated cells, but we don't necessarily have a full length of flagellum for our experimental cells. So we're going to rescale according to some length parameter L, and then only compare the length that we have. So let's say we rescale with L equals 60 microns, and our experimental cell, we've tracked 40 microns of tail, then we'll only compare the first 40 microns of tail and try and get a fit that way. And then the task really is to find the parameter set, so S, M, critical curvature kappa, proximal distal ratio rho, and length parameter L, that minimizes the sum squared differences between the experiment and simulation. And we're not going for a true minimization, but it's a minimization within the set of numerical calculations that we've done. So the closest fit that we have that we've already simulated. Okay, so we're going to compare with experimental data from a cell that we've imaged in a high viscosity fluid environment. So this is the cell that I showed right at the start. This is the experimentally tracked waveform. We can see it's approximately about just shy of 50 microns long. On the left, we have the mean absolute curvature along the arc length S, going from 0 to 3 times 10 to the 5. And we can do this fitting procedure, go through all our simulated data and find the best fit. And we get this simulation here. So you can see that the mean absolute curvatures fit pretty nicely, nicely on top of each other. And then in the bottom right, we plot the waveform for the simulation. This red piece on the end is essentially the free section that we haven't fit. So this section here, where we don't have data, where we essentially have the cell smoothly going to 0 curvature at the end of the tail to match that boundary condition. And I think you'll agree that this is a pretty good fit. Certainly, given that we haven't actually optimized the parameter set, we've just gone for the closest fit within the parameters that we've simulated. We can do the same for the cell in the low viscosity fluid environment. So this is the cell. This is the cell I showed you earlier. Same picture. You'll see there's slightly more asymmetry in this cell. This experimental cell actually had a bit of a head defect. So that's where the head was actually completely bent at the neck. So that's kind of induced more asymmetry than we'd see in a what we'd call a normal cell, but we'll see how well this fits anyway. And you can see the mean absolute curvature for the majority of the cell fits quite nicely. Hasn't done such a good job at the start where the bent neck is, and the simulation is reasonable, but not amazing. So there's some work to kind of think about how best to fit, but it's certainly doing not a bad job with characterizing the cell fairly well. So I'm pretty happy with that. So just to kind of sum up then, we've kind of taken these ideas of curvature control and kind of put together a simple active curvature model. And we've shown that this can really replicate many of the behaviors seen in sperm. And ideally, we've shown that it doesn't give us any weird behaviors that we don't see in sperm, certainly not for the parameters that we've calculated over. We've shown this really nice catastrophe bifurcation that enables switching between what I've called the low and high viscosity waveforms, which are just a way of talking about what we see experimentally in high viscosity fluids and low viscosity fluid environments. We've shown that the hyperactivated beats require an increase in critical curvature, and there's some idea about the fact that for these cells we get an increase in ATP, and this could potentially sustain this increase in critical curvature. That could explain that underlying mechanism. And we've shown that we can parameter fit to experimental data, and hopefully this might be able to give us some deeper insight into live cells and understanding what's going on, particularly with regards to kind of energy and fluid velocity, things that are hard to measure experimentally. So I'll just wrap up there and open up to questions. Some of the references of the stuff that I've talked about are here. Particularly, I'll point you to flagellarcapture.com, where you can find our FAST software, and GitLab, where all our code, or the majority of our code, is available open source and free for people to download and use. So thank you very much.